So thank you guys so much for having me on today. I really appreciate the opportunity to talk a little bit about some of our research. So my name is Abbie Bauer. I'm a first-year PhD student at the University of Delaware in the Interdisciplinary Neuroscience Graduate Program, which is really just a long mouthful way of saying I study neuroscience. Um, I work with Dr. Roxana Berchu in the Department of Kinesiology and Applied Physiology. And most of what our lab looks at is the effects of Parkinson's disease and healthy aging on the neural control of movement using neuroimaging techniques. So we do some functional imaging, some structural imaging. And what I would like to take some time to talk to you guys about today is one of our recent studies that we just published, which is titled Free-Water Imaging Unravels Unique Patterns of Longitudinal Structural Brain Changes in Parkinson's Disease Subtypes, which again is another bit of a mouthful. Um, so just sort of a brief overview in case anyone's unfamiliar with Parkinson's disease. It is a neurodegenerative disorder that affects the way that we move. And so some of the issues that we're facing as a field right now is that, as I'm sure many people are aware, we worldwide are experiencing an increase in the aging population. So we're seeing more people present with aging-related health conditions and concerns such as Parkinson's disease. And one of the key issues that we see in Parkinson's disease specifically is that it's a very heterogeneous condition. So a diagnosis of Parkinson's disease comes from a clinical diagnosis. So you would go to your neurologist and um, they would assess some symptoms that you're having. And Parkinson's consists of four cardinal symptoms. So the key one is bradykinesia, or that slowness of movement that we see. And the other cardinal symptoms are rigidity, so that inability to make a full movement of the joint, resting tremor or just tremor broadly, and then difficulties with posture, gait, and balance. And so, as you can imagine, it's very difficult to find two PD patients that look similar, let alone identical. And so this complicates when we start to look at clinical trials and research um, because people may not respond the same way to every type of drug or intervention. And so, in an attempt to sort of help delineate and better understand the heterogeneity, the field of research has put forward two motor subtypes that are categorized based on the ratio of tremor items to posture and gait items on the Movement Disorder Society sponsored Unified Parkinson's Disease Rating Scale, or the MDS-UPDRS, which is the third mouthful, I think, of this presentation. And so basically that's just a measure that patients will come in and their neurologist will have them like tap their thumb and their finger together, tap their toes, and just sort of assess their overall movement. Um, and so patients that present with a more tremor in their symptoms are nicely considered tremor dominant. And patients who present with more posture and gait and balance difficulties are what we would call PIGD, or posture instability and gait difficulties dominant, or that subtype. And so apart from just the symptom presentation, what we see is that these two subtypes have vastly different disease trajectories. So those who present with this PIGD subtype are more likely to experience non-motor symptoms, including cognitive impairment and a progression to dementia, as well as a faster overall disease progression. So they appear to deteriorate quicker than patients with a tremor dominant subtype. Um, and their symptoms, such as these posture and gait symptoms, are not always responsive to levodopa replacement therapy, which is kind of the gold standard in treating Parkinson's right now. So the bradykinesia and the tremor are much more responsive. Um, and so we don't have a full understanding of what makes these two subtypes so different. And one of the issues is in terms of like early stage Parkinson's disease, we can't really use traditional MRI measures. So part of that is because it is a degenerative disease, and we don't start to see these gross anatomical changes until the later stages of the disease. And so that's, like gray matter loss, things like that, atrophy, we can't really see them until later on in the disease course. And the areas that do change or potentially change earlier in the disease course are some of these subcortical structures in the basal ganglia that are not always visible on standard T1 images. So those would be, like the globus pallidus, the subthalamic nucleus, the substantia nigra, These regions are very hard to visualize on a standard T1, and they're very tiny, which makes them difficult to sort of draw any conclusions on or run analysis on using a T1. And so something that's relatively new in the field is this use of diffusion tensor imaging. And so that is a new type of, not new, but a newer type of structural imaging that looks at the overall diffusion of water molecules within one of these voxels in the image. And so specifically what we used here is free water, which is one of these DTI measures, and it is sort of a measure of the extracellular water. So not inside of the cell, but what's around outside of the cell. And we've seen that this value increases, generally speaking, in neurodegenerative disorders, including Parkinson's disease, which is why we chose it for this one. And it's been shown to increase over time in clinically relevant regions in this condition. And so the goal of this particular study was to utilize free water to help us track these longitudinal changes in these PD subtypes over 2 years in a group of patients from the Parkinson's Progressive Marker Initiative, or PPMI, which is a phenomenal longitudinal study that's been put together by the Michael J. Fox Foundation. And it enrolls participants who are newly diagnosed with Parkinson's disease and sort of tracks them over, I think, as long as they're willing to follow up. I think they're at like 11 years now or something. So it's just like a massive database that's available to researchers. It's a really awesome tool. Um, and so specifically for this, we hypothesized that compared to tremor dominant, PIGD patients will experience a faster rate of change in free water, and that they'll experience these changes outside of the basal ganglia. And part of the reason that we thought this is because these symptoms that they experience, these non-motor symptoms, these balance symptoms, are not responsive to dopamine in the same way that the tremor symptoms are, and that they might be related to structures outside of the basal ganglia that help to control these movements. So our participants here, like I said, we got them from the PPMI database. We had 28 tremor dominant and 18 PIGD patients. They were well matched at baseline on age, sex, total UPDRS score, which is that overall sort of clinical score indicator, as well as cognitive status. And it just so happened that they were all well matched on these baseline free water measures as well, which was a kind of a happy coincidence. Um, and these were all newly enrolled de novo, so non-medicated patients at the time of their enrollment, so their baseline. And so we've got two cohorts here, but really cohort 2 is just a subset of cohort 1 that has that motor score at year 2 off medication. Not all participants had that, so we sort of had to pare it down if we wanted to run any type of additional analyses. So that's where you'll see that cohort 2 come in. So the regions of interest that we picked for this study were some of the basal ganglia nuclei, which you can see at the top there. And in terms of the non-basal ganglia nuclei, we also looked at the thalamus, the PPN, red nucleus, lobules 5 and 6 in the cerebellum, as well as the vermis and the dentate in the cerebellum. And again, that's because we thought that the PIGD patients may have changes outside of the basal ganglia. So we wanted to take a look at that because it's not necessarily fully understood in Parkinson's. So what we found was that the PIGD patients did have a greater increase in free water in the putamen, the globus pallidus, and in cerebellum lobule 5. Um, and so you can see that here that it's not only are they increasing in the free water value, they're increasing faster than the tremor dominant patients do. And so that held for the putamen and the GP in both cohorts. And then in that cohort 2, which is just a subset of cohort 1, we can also see that in lobule 5, they did have a faster increase in free water values. And so kind of putting this back into context, the putamen is the input structure of the basal ganglia, and the GP is the output structure, broadly speaking. And so if they're experiencing changes in these regions, it might help to explain why these PIGD patients experience more severe motor symptoms when we compare them to TD patients. And so it might just sort of be a sort of start to understanding why these patients have such a different trajectory. And then in terms of the cerebellum, lobule 5 is involved in sort of motor planning, coordination, and execution of these motor plans in tandem with the motor cortex. And so we don't fully, as a field, understand the role of the cerebellum in PD. There's been some new studies that have come out looking at like connectivity and functional changes. And we're seeing differences, but it's not fully understood yet. But we suspect it might be related to some posture and gait deficits in PIGD in this case, or again, in terms of those poorer motor outcomes. Um, and so just sort of like the take home from this study, since it's a bit lot if you're not a PD researcher, we found that the patients who present with predominantly posture and gait symptoms had a faster rate of free water increase in several motor regions, both inside and outside of the basal ganglia over 2 years. And really, this helps to set the foundation for the design of future subtype specific interventions. As I said earlier, patients may not all respond to medication or interventions or exercise interventions the same way. And so if we take this heterogeneity and this potential difference in pathophysiology into consideration, it might help to explain some of the fact that these clinical trials don't necessarily yield positive outcomes and help to design better ones in the future. Um, and so I would like to thank, obviously, Dr. Roxana Bertu, who is the PI of the lab, as well as Dr. Chung, who is our postdoc, and Sophia, our master's student, as well as Dr. Justin Martello over at Christiana Care Neurosciences, who's been a fabulous collaborator, and the Michael J. Fox Foundation and PPMI for making this data available to us. So yeah, thanks again so much. And if you have any questions, I'd be happy to answer.